thank you all for coming. It's a pleasure to be here. What I'd like to do today is tell you about the work that's going on principally in my laboratory on stem cell biology as pertaining to the cardiovascular system. This is the overview of what the lecture is going to look like. We'll talk very briefly about the problem of myocardial infarction, and then really jump into some stem cell biology, talk about the biology of stem cell differentiation, some work that my lab is doing on how transcription is regulated during differentiation, then go to more applied aspects. We'll get progressively more applied as the lecture goes on, talking about stem cell therapies for cardiac repair, and then end up with tissue engineering approaches and some closing thoughts in the end. People who know me know that I have very bad seminar manners, and I'm always interrupting speakers and not allowing them to finish sentences or trains of thought or anything, and so I expect to be treated no better when I lecture myself. So if people have questions, I'd invite you to just speak up, please, along the way. It makes it more interesting. Okay, so here's the fundamental problem. Ischemic heart disease remains the #1 cause of death in the United States, despite major advances in treatment, and then the downstream consequence of myocardial infarction, congestive heart failure, remains the #1 cause of hospital admissions in patients over 65. So it's a huge public health burden. This is the anatomical substrate that really causes myocardial infarction. This is an atherosclerotic coronary artery that's had a rupture of the fibrous cap. The blood has come in contact with the highly thrombogenic necrotic core, and it caused an occlusive thrombus, and this is what has killed this particular patient. Now, patients who are able to survive the myocardial infarction in its acute phase go on to, excuse me, a sort of a stereotypical set of healing events. Now, the heart is arguably the least regenerative organ in the body, maybe vying with the central nervous system for this dubious distinction. People argue, is there no regeneration, or is there just very, very little regeneration in the heart? But the simple fact of the matter is that the heart's not up to the job of repairing itself after a myocardial infarction. And so rather than generating significant amounts of new muscle tissue, the body goes through a phase of granulation tissue formation that then converts into scar tissue. So this is an autopsy specimen. You can see this patient had an infarct in the anterior lateral wall associated with a left anterior descending coronary occlusion. Here's the normal myocardium in the back wall of the heart. And if you follow, it's hard to do it with this arrow, but the endocardium is right along here. So this is a thin wall and an aneurysmal dilation, marked expansion of the left ventricular cavity. So this is the anatomical substrate of heart failure, if you will, a dilated, ballooning out heart that loses its mechanical efficiency, and it progressively spirals downward toward heart failure. So this is what we would like to prevent from happening by harnessing the power of stem cells and applying them in some manner. I like this diagram, not because anybody can actually make any sense out of it, but first I've sat through so many talks on signaling pathways that I like to inflict my own kind of punishment on people who do that sort of science. This is a diagram that was redrawn from Gilbert's Developmental Biology textbook, and it really is the fate map for many but not all of the tissues in the body. And the notion is that you start, we all began life here as zygotes, go along the way to be sort of an inner cell mass, a pluripotent stem cell. This is kind of the equivalent of an embryonic stem cell. And all the way down here, you go through a gastrulation, you make mesoderm, different types of mesoderm, and way down at the bottom, you can make cardiomyocytes. So if you think about starting with something like an embryonic stem cell, which is essentially the equivalent of a cell in the inner cell mass of a blastula, you have, you at least need to make mesendoderm, then you make mesoderm, then you need to make lateral mesoderm, splanchnic mesoderm, and on to cardiomyocytes. So there's multiple decisions that have to go along the way. Cells have all manner of choices that are open to them, and the trick is how do you get them to take the choices that you want to the exclusion of the choices that you're not interested in. I like this cartoon because we're going to fix the heart. The heart needs cells. Let's put cells in. Really, what could go wrong, right? Well, turns out that if you take a cell that's as potent as an embryonic stem cell and do what most people in the field have done, myself included, just squirt them in and hope for the best, thinking Mother Nature is going to take care of you. This doesn't work so well with embryonic stem cells. What you end up with is something that looks like this. This is a mouse heart where we transplanted in about 500,000 undifferentiated embryonic stem cells. You should see a ventricular cavity in here somewhere, but what's happened is, at least at this level, much of the heart has been replaced by a complex tumor. You can see it's got cystic elements in it. And under higher magnification, we see ectoderm derivatives like keratinizing squamous epithelium. We see mesoderm derivatives like cartilage. We see endoderm derivatives, sort of gut-like structures with goblet cells. So this is a classic teratoma. The heart doesn't have the necessary instructive signals to guide these potent cells towards the tissue-appropriate fates. And so they just do whatever they want and make and this would, of course, be a therapeutic disaster if we were to take some poor patient who had a myocardial infarction and give them a cardiac teratoma. So if somebody wanted to tank the field of cell therapy, this would be the fastest single way to do it. So this brings us to the question is, how is it that we're going to direct the differentiation of the cell? I view this as sort of like a game of roulette. There are many, many different cell fate choices that an embryonic stem cell can have. Depends a little on how you define a cell type, but there's, I think most people would agree, over 200 different cell types in the human body. And for therapeutic applications, most of us would like to have one or just a few specific desired cell fates. So how are we going to do this? Well, if we redraw some of the information that was in the Gilbert pathway diagram that I showed you previously and superimpose on that important information that's been gleaned from the world of developmental biology, we actually have some important information that tells us how to direct the differentiation of cells. And one of the things that I've been impressed with is how useful developmental biology has been as a guide to directing the differentiation of embryonic stem cells. So you start out as a cell of the epiblast or an embryonic stem cell equivalent. In terms of making mesoderm, we know the major signaling pathways that are involved are the Wnt pathways and then members of the TGF-beta superfamily, particularly bone morphogenic proteins and activin A. And if you activate those pathways, you make what's called initially mesendoderm that subsequently forks to definitive mesoderm and definitive endoderm. Conversely, if you inhibit those pathways, the default pathway of most of these cells is to make ectoderm. So paradoxically, even though the central nervous system is one of the most difficult tissues to regenerate in the body, it's probably the easiest stuff to grow in a petri dish using embryonic stem cells. Because if you just leave the cells alone and ignore them, they make tissues of the central nervous system. And then we go on down the way to make precardiac mesoderm and eventually cardiac progenitors. And then the bottom line is not to remember all these different molecules, except that we know a lot about how the embryo puts itself together from careful work that the developmental biologists have done. So harnessing some of this information, we've actually gotten quite good, and this is not just work that's been done in my laboratory, but many laboratories around the world have been working to direct the differentiation. And if you take a sort of a spontaneous differentiation approach, say take cells in a three-dimensional clump called an embryoid body and treat them with serum, using human embryonic stem cells, this is quite inefficient. You get on a good day maybe 1%, typically about 0.1%. This is shown here, little clumps of cells stained with myosin heavy chain antibodies. This actually exaggerates the frequency of it because, again, it's usually quite low. Conversely, when we do a directed differentiation treatment by taking developmental pathways and activating them with recombinant proteins, we can get pretty good numbers of beating human cardiomyocytes. In this particular case, it looks like maybe a third of the cells or so in the field would be human cardiomyocytes. And once you have that, it's easy to do something simple like a density gradient, take advantage of the fact that these cells are fairly dense, and purify out a lot of the contaminating cells and get cultures that are on the order of 90% or so human cardiomyocytes. And when you get an experiment that works well, it's really quite dramatic. This is a video that we took, and you can see there's beating human cardiomyocytes. One cell would be about that dimension. So the things that you see that are really bright are three-dimensional little balls of cells. We call them cardioballs. They're actually viable cells sitting on top of other cells. And these things are all networked together. They form junctions that are kind of like intercalated discs. They've got adhesion proteins and they've got gap junctions. And the whole network will beat synchronously under the microscope. And we've got dozens of petri dishes with this kind of stuff that's beating merrily away at about 70 beats a minute in the laboratory right now. So in theory, it's just time and money right now. You can make vats of human cardiac muscle if you simply have the resources to do it, because the cells can be expanded infinitely. And if you don't mind spending all of your money for recombinant proteins and human labor, you can generate large numbers of cells for therapeutic purposes or experiments. So we're interested in both fundamental questions of cell biology and in sort of applications of stem cell technology. And one of the fundamental questions we're really interested in is, what is it that makes a cell, a differentiated cell state stable? I view this sort of as a thermodynamic well into which a cell falls because of some positive feedback loops that it gets into once it gets to a differentiated state that is somehow favorable. And so my notion, so what is it that stabilizes this? That there's some kind of dynamism between chromatin regulation, transcription, translation, things like that, that the cell is happily resonating in, and that gives stability and positive feedback loops, and that is what allows us to have something that we call a cell type. And so one of the things we're looking at is what's the role of chromatin and transcriptional regulation and translational regulation in defining differentiation pathways. So the first part of this work is something that a MD Ph.D. student in my laboratory, Jonathan Golub, has been working on, looking at chromatin dynamics and transcription. And so this is the differentiation scheme shown down here. We initially treat the cells with activin A followed by BMP4, and by about 2 weeks later we get beating cardiomyocytes. OCT4 is a transcription factor that's characteristic of undifferentiated pluripotent cells, and you can see OCT4 levels plummet nicely as the cells differentiate. Conversely, there's a transcription factor called brachyury that's characteristic of mesoderm cells, and so that spikes up. You see we get on this particular experiment about 30-fold induction of brachyury, and then it crashes back down as the cells go on past the mesoderm state to more definitive cardiomyocytes. Conversely, the red-brown line you see along the bottom is a transcription factor called SOX1 that's characteristic of neuroectoderm. This is the pathway that we wish to suppress with our treatments, and that's flat all the way across. So what this tells us, and it's shown just by this little arrow on the right, that we think we're biasing our cultures towards mesoderm and endoderm with these cytokines that we're treating, and we bias the cells away from an ectodermal fate. So we think this is truly directed differentiation, not simply expanding through proliferation or survival advantages. So, if you can make these cells, what's it good for? Well, I think this is such a fundamentally cool process that you can use it to answer lots of basic questions in molecular biology, and I'll just give you one example about that. People who study transcription have been doing some very interesting things lately, and that have taught me at least some surprising things about the biology of transcription. What you're looking at here, this is a result of some experiments where people did what was called a nuclear run-on experiment. They took RNA that had been very recently synthesized and they labeled it with a pulse label approach so that we know that we're only looking at stuff that was very recently made. So this is a kinetic analysis, not a steady state analysis. And then they sequenced everything that came out from a whole population of cells, and they gave you just an average look at what a single gene would look like. This is called metagene format. And what you see, and so the red part is conventional transcription going from 5' to 3', and the blue is stuff that's going in the other direction. And some surprising things came out of this. So what's the profile of the average gene? The average gene, if we look at the transcriptional start site, there's a big burst at the 5' end of it. And this is the process of RNA transcriptional initiation. And then this is the part that is RNA elongation. So what that tells us is that the cell is really no different than the rest of us. It initiates tons more stuff than it ever finishes. And I found that to be just fundamentally interesting, I guess, because I never had a clue about that. The other thing that's interesting, and I have no idea what this means, is that when cells initiate transcription, they do it in both directions. They do it 5' to 3' in the sense strand. They also go 5' to 3' in the antisense strand, in the opposite direction. It doesn't carry on as robustly, so there's not the elongation in that direction. Nobody has a clue what that means at this point. But we were interested in this fact, why is there all of this transcriptional initiation that is much in excess of elongation? Well, that was an average gene, that if you took many thousands of genes and collapsed them into one. What if you look at an individual gene? Do they actually look like that? Well, no, that was a mathematical average and sort of an artifact in that sense. So what you're seeing here is, in red, is an actual gene. It doesn't even matter what it is. But what they found is there are two classes of transcription that are going on. One where there's initiation and pretty much efficient read-through from start to finish. And that's the way most of us, at least I, had thought about transcription as being initiated. You start it, once you start it, you finish it all the way through. The big spike came from genes that look like this, which is what they called pause transcription. Pause is not really a good name for it, but what that means is that this is lots of initiation and very little follow-through. So the enzyme complex that's making the RNA has to be falling off the DNA template and starting over again. So it's kind of spinning its wheels in the process of it initiates but doesn't follow through. And this is maybe a third of the genes in the cell are doing this. That seemed very strange to me. You can imagine all the energy that it would require to make short little segments of RNA and never follow through on them. So the question that we wanted to know is, why would Mother Nature do this? Why would it do something that seems so energetically inefficient? Well, the models that people have built, there's a collaborator of ours named Rick Young who's done some very nice work on this. But in order to understand, I have to tell you a little bit about some tools that they've developed using chromatin analysis. You remember chromatin, of course, is DNA plus the proteins that it's wound around. The main proteins that are involved in chromatin are called histones, and we won't fuss too much about what the different histones are, but histone H3 turns out to be a major player because there's a lot of post-translational modifications in histone H3 that determine how accessible the DNA is to transcription factors coming in and making RNA from it. So if we look, here's the transcriptional start site for a gene, and the first thing that happens is that this is broadly there's a post-translational modification on histone H3 where it's methylated on residue lysine 4. So trimethyl lysine 4 on histone H3 is a mark for transcriptional initiation. It turns out there's several other marks, but that's going to be the one that we see. So what the model is RNA polymerase lands after the histones are modified. It starts making RNA. RNA polymerase is also phosphorylated. That's what that ball means. And when it reads all the way through, as you can see, there's this full-length strand with both 5' and 3' components to the transcript. And eventually, obviously, so the 3' transcript means you've read all the way through. And if you have only 5' parts of the transcript, that means it's initiated but hasn't elongated. So in the other model, if you have the trimethyl lysine 4 on histone H3, RNA polymerase can initiate. It starts making a short transcript, but it's got no 3' transcript, and it doesn't have some of these other secondary post-translational modifications. We're not going to get into that for the purpose of today's lecture. So the bottom line is, how do we use information like this to tell us what the cell's doing? Well, we decided to condense this and simply use two things. We're going to say, is it making a full-length transcript by having the terminal ends? Is there a 3' transcript? That tells us that it's full-length read-through. And is a particular gene having this particular histone modification of trimethyl lysine 4? So that lets us tell whether it's initiated and reading through or initiated without reading through. So we did this genome-wide in embryonic stem cells. And the reason that we did this was to answer the following question. My notion was that this non-productive transcription, that is actually not a cell wasting its time, but this is actually a transition state for genes that are either going to become active or going to get shut down. And it's actually an important regulatory mechanism. The rub is that everybody who had been studying this so far had looked at cells that were in steady state. They were just boring fibroblasts sitting in culture, or less boring cells, but just sitting at a particular state. So we needed a cell that made a dramatic change in state in order to test the hypothesis. And of course, we had embryonic stem cells that were doing something dramatic, so we put them to the test. The way we did it was using a technique called chromatin immunoprecipitation to get down the parts of the genome that were associated with this modified lysine on histone H3. We got the DNA regions of interest, and we simply hybridized it to whole genome arrays that had segments from about upstream about -4 kilobases to downstream about 2 kilobases of the known transcriptional start sites for all known genes. So we had about 25,000 genes that were being interrogated this way. So what do we see? Well, let me just, obviously I can't show you 25,000 genes. I'll show you 3 that will sort of tell you what things look like. And what we found is that this gene brachyury that makes this transcription factor that marks mesoderm, it starts out in what we'd call this non-productive state. If you look at the chromatin, it's got the lysine 4 marks around the transcriptional start site. This is going both in the sense and in the antisense directions. And it doesn't change much from undifferentiated to differentiated. But the RNA spikes up, and this is actually underrepresentation just because the arrays tend to squelch the signal. But there's really quite a dramatic bump up. So this is an example of gene activation going from non-productive to productively transcribed. Some genes just hang around, don't do much change. This is NKX 2.5. It's a gene that's going to be markedly upregulated 2 weeks later when they become cardiomyocytes. But this gene was interested. This is called NeuroD1. It's involved in neuronal differentiation. And remember, this is a pathway that the cells are going to suppress because they're not going to become neuronal cells based on our treatments. So this is initially in sort of an optimistic state. It's got initiation, but there's no transcription at the 3' end. But look at how markedly downregulated this histone modification is later on. So this goes from being sort of idling the engine, if you will, to turning off the car. It shuts it down. So if you take that, so if we look at genes kind of in this picture and ask what happens as the cells differentiate on a whole genome level, you get a picture that looks like this. Here's what the portfolio of the cell looks like when it's undifferentiated. Oh, maybe a third of the genes are productively transcribed. Half of them probably are non-productively transcribed. Look, that's just huge to me. And then about a small 10% or so are completely silent. And if you just look at the whole portfolio genome-wide after differentiation, it doesn't really look any different. But this doesn't mean that there's no changes going on. It just means if there are changes, they must be offsetting. So if you look at what percentage of genes actually change, we had a debate in our lab as to whether there would be a bunch or a little and what a bunch or a little would actually mean. It depends a little bit on what criteria you use to define a bunch or a little. Bottom line comes out to 12 to 18% of cells, of genes actually change their regulation state, depending upon what you what you call. We took an informal survey around the building. The molecular biologists thought 12 to 18% was huge. The cell biologists were sort of singularly unimpressed with this. So it's it's whatever you happen to think 12 to 18% is, I guess. What's going on as these genes are changing their state during differentiation? I guess so. The question was, is there a key step as we go from DNA to RNA to protein? What we've shown previously in the study was that as you go from RNA to protein, about 90% of the genes just go on and get directly translated. There's not a subsequent regulatory step. But about 10% go through a pause state where the RNA sits around for a while until there's a particular cue that says, okay, go ahead and translate and make protein. So the hypothesis was maybe something like this is going on during transcription as well, that you can initiate, but you don't elongate. So the question is, of the genes who change their state, what fraction hop straight from inactive to active, and what fraction go through this paused intermediate? And that's just shown up here again schematically. Do you go through this non-productive state as a transition state, or do you bypass it and jump directly? And we did two different biological replicates, and we got an astonishing answer, at least to me. 98 to 99% of genes who change their regulation use this model on the left, where almost everything seems to go either to or from the non-productive state. It looks like it is the transition state that cells use as they change their transcriptional regulation during differentiation. That most people seem to think is high. Couldn't have been a lot. We probably missed a few by just because we waited 2 days. This was a 2-day in-between period, so we could have even missed a couple who changed from from red to green in the middle. So this is just an example of how embryonic stem cells can tell you something fundamental about cell biology, and I think this is probably true for cells in general as they change their state. They use this non-productive transcriptional regulation as a key intermediate point to either go up or go down. And that's the model basically, but I think we've already talked about that. Why would cells do this? I think it makes the cell poised so that it can more readily adapt to changes in its environment, so that you get a cue that says it's time to make some kind of change. If you're already initiating transcription, to then go on and do full elongation, it would probably take you much less time than it would to unwind the chromatin and make the DNA accessible. So that's my view is that this is kind of like idling your car. You're sitting there, you're ready to go as soon as you get the signal, as opposed to having it turned off and parked somewhere.
Well, so Chuck, do you think, now I'm going to interrupt you, do you think that this is a model in general for all types of cells, or is this something unique about a cell that has lots and lots of different decisions to make? In other words, if I'm a cell that knows it's going to end up being a red blood cell, for example, does it really need to have this kind of poising to act?
So I have no data to address your question. Oh, sorry. So the question is this non-productive transition intermediate, is it unique to embryonic stem cells, or is there something specific? Is it a more general model when cells may change their state? And we have no data. My bet is that all cells are going to do this. We know all cells are doing this non-productive transcription for about a third of their genome. Why would you do this? If there weren't it must be tremendously energetically costly. Why would you do this if there isn't a purpose for it? And so my bet is that if you cytokine treat a cell, that you will see that the genes that change their expression pattern are going to change as well. But we need to test this with other cell types, obviously, because it could just be a special cases for an ES cell. Any other questions? So this is sort of the end of the fundamental science question. We're going to start to get a little more applied and disease related here. And this is some work that Mike Laflamme, who many of you may know, started when he was a postdoc in my lab, and we've continued on a collaborative basis. And the question is really, since we know how to make large amounts of human cardiac muscle, can we use them for anything therapeutically? We've done a lot of work in transplantation. This is the preferred host. This handsome fellow is the athymic rat, who isn't quite as nude as the nude mouse. He's sort of the alopecic rat. But they turn out to be very good hosts for human cells. And we spent a long time learning how to make our cells survive after we transplanted them. It took us about 2 years to, everything we put into the infarct died. Turns out a myocardial infarction is a toxic environment for myocytes. That sounds like duh. But it took us 2 years to learn this. And we developed a cocktail of prosurvival factors finally to keep our cells alive after we transplanted them. And once we did that, we were able to grow decent amounts of human heart muscle in these experimental infarcts. So the question is, did it have any impact on physiological function? So we infarcted these animals and then came back 2 days later, did a baseline echo to see how dysfunctional their hearts were, and then came back 2 days after that and engrafted the cells. And the notion was to give the heart a few days to let the inflammation subside and let blood vessels grow in through granulation tissue so we'd get better graft survival. We put in 10 million of these directly differentiated human cardiomyocytes or control cells or other vehicle controls and so forth. And we waited a month, and we looked at the animals physiologically and histologically. Just to jump to the histology shown on the bottom here, we got decent size amounts of human myocardium. You can identify human myocardium by different gene expression patterns, and it's morphologically quite distinct because it's fairly immature looking. And so this is human myocardium here shown with beta myosin, the principal myosin of human hearts. This is a rat myocyte over here. It's troponin positive, but beta myosin negative. Sometimes we saw nice close apposition in what looked like intercalated discs between our graft cells and our host cells. More commonly, there's a band of scar tissue that separates the two, and so that's a problem. And what you see here is a couple of cells in mitosis, so these are metaphase chromosomes, and so these cells proliferate, and they're proliferating a month out even. So we get a break from Mother Nature because compound interest lets our grafts expand about 7 to 10-fold. So physiologically, what do they do? The way we assessed this was through echocardiography and through magnetic resonance imaging. This is an MRI using actually a human 3 Tesla scanner that we have at South Lake Union. And this is a rat heart. So this is a heart about the size of your thumbnail. But you can see it's got all its parts. There's the left ventricle here, right ventricle here. You can see the great vessels coming out for the outflow and so forth. So this is the best way to get structure and function relationships in the rodent heart because of the high resolution that MRI affords. If one can afford to pay $200 an hour for magnet time, that is. So what do we see? What we see is that these hearts, if you look in the infarct region in particular, there's substantially more systolic thickening. And that just means like you flex your muscle and how much does it thicken. And in the non-infarcted regions, the wall thickens about 55% with each beat. And you can see that the animals we put our human cardiomyocytes in had about 2.5-fold better systolic thickening than any of our control groups, including those receiving irrelevant cells or various vehicles. So not all the way back up to where the non-infarcted region is, but still a substantial protective effect. So we were very encouraged by this. Now, it's probably more clinically relevant to put this into a model of established heart failure because I think nobody at this point would be willing to approve a clinical trial in patients who don't have established heart failure. Because again, embryonic stem cells are too experimental. And so we asked the question, could we put human cardiac muscle into animals with established infarcts and a month's worth of heart failure or so? And this is a good news, bad news story. The good news is that the cells engraft like crazy. This is a band of, shown on the right, this is a band of beta myosin positive human muscle that formed in this rat heart. So they grow very well. They actually find it a very hospitable environment to colonize. And so we were encouraged by this. We went on to do the physiological studies. Before we get there, just interestingly, we also formed a lot of human microvessels in this particular instance as well. This is human CD31 identifying human capillary networks. But what we found when we went on to do physiological studies was once you have heart failure established, you can grow all this human heart muscle back in the heart, but it didn't budge cardiac function. We had all these different control groups that I told you about before, but it really doesn't matter which group is which. whether we look at ventricular dimensions at end diastole and systole, or fractional shortening, which is a linear equivalent of ejection fraction. So this is a systolic functional index. Didn't make any difference. Bottom line, I think it's easier to prevent heart failure than it is to cure heart failure. So I think this has some clinical implications perhaps for the patient population that we may want to think about targeting with a cell like this. Okay, so for the last part of my talk, what I'd like to do is tell you about the work that we are doing in tissue engineering. The basic concept of tissue engineering is really to take cells and make three-dimensional masses of tissue in a petri dish, commonly done by seeding cells onto a three-dimensional porous scaffold. We spent a lot of time working on porous scaffolds, but they have certain problems. They cause a foreign body reaction, for example. As if they're biodegradable, there's oftentimes acidic degradation products that may be toxic. And so a grad student in the lab, Kelly Stevens, whom you see in the upper left corner, Kelly stumbled on a technique to do tissue engineering in a scaffold-free manner. And what she did was she was just playing around in the lab one day and made a big batch of human embryonic stem cells and put them into a petri dish that the cells couldn't stick to, and put that on an orbital shaker in the incubator. And we thought we'd make a lot of little balls of cells. But instead, because of whatever hydrodynamic reasons, she made a disk of tissue. They aggregated in the center, and they formed a three-dimensional disk of human cardiac muscle. So you can see it here on the major plane, parallel to the major plane of section with an H&E. It's kind of boring to look at it. It's round, and it's reasonably thin. But the cool thing is, if you look at it in real time, you start making these chunks of beating human heart muscle. So, and this is probably about, you can make them from anywhere from a few millimeters in maximal dimension to centimeters. It's just a question of the input number of cardiomyocytes. And they all tend to be about 300 to 400 microns thick. That's key, because if you get too much thicker than that, you start to have a necrotic core due to diffusion limitations. So, what can these cells do? Well, they could be useful for physiological experiments. We wanted to know whether they were electrically coupled, for example. So we loaded them up with a calcium indicator. And so this turns bright green whenever there's a systolic rise in calcium. And what you see is over here on the right-hand side is where the pacemaker is. This is a spontaneously contracting patch. And then a wave of calcium sweeps across the cells as they depolarize. And so it tells us that they're all electrically coupled through gap junctions. And if we put the gap junction blocker heptanol on it, this completely shuts it down, and the cells will just fire autonomously. And we can quantify this using line scan mode and confocal, but it just tells you what your eye says here, which is basically when these guys all go together once there's a spontaneous depolarization. So that was kind of cool. We've been working on looking at the mechanics of these cells, and this is some initial work to that end using video edge detection software to monitor contractile function. These are unloaded contractions. And so we just started stepping up the rates. You could see here at every 2 seconds or half a hertz, then 1 hertz, 1.5, 2 hertz, et cetera. And interestingly, we started to get incomplete relaxation as we went up and up. The constructs would kind of ball up as I don't think it's the cells being incompletely relaxed. I think they're just the whole three-dimensional mass of the tissue. And if we loaded them, they'd probably relax just fine. But you can see they capture well at a half a hertz. They capture well at 2 hertz. But if we start doing them at 4 hertz, which is really quite rapid, so 4 beats a second, these cardiomyocytes can't go this fast. So you start getting very irregular beats and some arrhythmias. And that's quantified here, that all these constructs that we made capture well out to, say, 120 beats a minute. When we try to get them to go to 150 or 180 beats a minute, we start to lose them. But that makes sense. The fetal heart only can go at about a maximal rate of 150 or so, and human cardiomyocytes have certain physiological limitations. So we wanted to take these and transplant them and see if we could make more human heart muscle by a tissue engineering approach than we could with a cell therapy approach. And we expected to see much better survival when we did this, because we wouldn't be putting the cells through all the stresses of enzymatic dispersion, squirting them into a tissue with no attachments to one another, that sort of thing. And what we saw, unfortunately, was much worse survival. So this was transplantation into a skeletal muscle model, actually, which is a much faster way to crank through things than doing open heart surgeries all the time. So this is the host skeletal muscle over here. This is a stain for the beta-myosin that you find in the human cardiomyocytes. And what you find is if 1 week after implantation, there's a few scraggly looking little cells and a lot of inflammation. And so really what's happened is we put this patch in, it's all died off, and it's mostly being eaten by macrophages at this point. So that was disappointing, and I should emphasize we did this using our pro-survival approaches where we'd spent years developing, and they seemed to be woefully insufficient to make an impact. So we concluded that this was poor survival. What next? Well, why was it that the cells were living and the patches were dying? The obvious difference was one of geometry, and if things that are little clumps of cells can live on diffusion, whereas a big three-dimensional patch is going to be much more diffusion limited. So we thought it was probably an ischemic hit. If that was true, we thought if we could find some way to hasten vascular ingrowth to these things, this should accelerate perfusion of the constructs. And this is where I was really impressed with the work that Kelly did, because she really figured out most of this stuff on her own. So the goal, I said, okay, Kelly, just go off and grow some blood vessels into these things. What we need is like a plug-and-play network where just make these prefab vessels, slap that baby in, the host will grow in, they'll connect, and this will be fine. And remarkably, she actually goes off and does this. So the top panel you see is an endothelial marker, CD31. And with these cardio-only patches, there's really no CD31 positive cells, lots of beta-myosin positive muscle cells. The first thing we tried was just doping in an endothelial population, a standard laboratory line, human umbilical vein endothelial cells. This did not work well at all. We threw those things in, and all they did was make little dead endothelial balls at the center of the patch. So you can see these little brown, round things. They're endothelial cells, but they're all dead, and they're all at the center of the patch. Nothing morphogenic took place that looked like it was the organization of a vascular plexus. So what Kelly did next was reason that they were missing something that was the equivalent of like a smooth muscle cell or a pericyte, something to crosstalk with the endothelium and provide some structural stability and maybe some back and forth crosstalk. And lo and behold, she added, in this case, mouse embryonic fibroblasts, which are highly potent in terms of secretory activity, and she got something that looked very different. And I'll show you at higher magnification here. These are human endothelial cells that are starting to form a network. Now, I wouldn't tell you these are blood vessels, right? This is something that is sort of reminiscent, though, of an early vascular plexus in an embryo that's just forming. So we thought that maybe we were on the right track with this particular approach. So we needed something that was going to, excuse me, that we needed this crosstalk between these two cell types in order to make something looking like a vascular network. So Kelly took these and went back to this model of implanting them into skeletal muscle. And this time, and this is sort of one of the representative examples. I won't show you just the best one, but you can see we have readily detectable amounts of human cardiac muscle that survived. And interestingly, this is human CD31, so there are little human capillaries that are present throughout this tissue. And if you do quantitative morphometry to ask what's the impact on survival, we got a huge impact, about a 12-fold increase in graft size by pre-vascularizing these things with the human umbilical vein endothelial cells and these mouse embryonic fibroblasts. So again, this suggested to us we were on the right track, and there was probably something to this notion of pre-vascularization. So the next goal then was, human umbilical vein endothelium. Nobody would want that as a therapeutic cell, and mouse embryonic fibroblasts, of course not. That's out. So can we find something that is relevant to replace these funny cell types that we did this first experiment with? Well, at the same time, a graduate student in the laboratory, Marilyn Norse, was working on deriving endothelium from human embryonic stem cells. And again, a lot has been taught to us by the developmental biologists in terms of the pathways that create endothelium. And we knew that if you just took a three-dimensional embryoid body, like you see in the upper left, and stain it with endothelial markers, you occasionally found these areas that looked like little cords of endothelium that might be forming something that would be useful and maybe the rudiments of a blood vessel. So the cells do it on their own, but it was inefficient. Is there a way that we could goose the process up in some way? So Marilyn screened a bunch of different induction regimens, and I won't spend a lot of time going into the gory details on this, but vascular endothelial growth factor, VEGF, turned out to be the cat's pajamas in terms of inducing endothelial formation from human embryonic stem cells. You can see using CD31 in Western blots. These three lanes are all biological replicates. You can see massive induction of CD31 compared to a variety of other different interventions that we did. And since we like CD31 because it's a surface epitope that you can use then by flow to pull your cells out. So we said, okay, let's take this approach and treat our cells with VEGF to induce endothelium. And now, are these really endothelial cells? Of course, CD31 can be expressed in lymphocytes and monocytes and things like that too. So we wanted to be careful that we weren't getting tricked by hematopoietic elements. So a more definitive marker for vascular endothelium is a cadherin molecule, VE cadherin, that's highly endothelial specific. And you can see we get good superimposition of vascular endothelial cadherin with CD31. Shows reasonably well in the merge here. And shown for people who like, Dan Sabbath always likes flow cytometry. So we can do this by flow cytometry. Most of the cells turn up in this double positive gate for CD31 and VE cadherin. And it on average, we get 4 to 5-fold enhancement of endothelial cell yield. Some runs we get 5%, some runs we get 15%. You always see some run-to-run variability with stem cell differentiation. But it looked like we had a tool for making endothelium now. Well, what about the mesenchymal cells? Well, another cell type that's under clinical trials right now are the merostromal cells, also called mesenchymal stem cells, MSCs. We just call them MSCs because no one can agree with whether they're stem cells or not. And if you take mass cultures, we found that they work just as well as the mouse embryonic fibroblasts, maybe even better, at forming things that look like vascular channels and these chunks of engineered human myocardium. And then for doing some mechanistic studies, we've got subclones out of these mass cultures that have been immortalized, and we found one that works really well and one that's really poor. And so the notion is now, so this is the one that works better on average than the bulk cultures of, over here, the bulk cultures of the mesenchymal stem cells. And then we've got one that works much worse. So that probably explains why there's lots of noise and heterogeneity with these MSCs. Anyway, we've got a system to sort out what the principal signals are that are responsible for this crosstalk between endothelium and mesenchymal cells. Okay, so to finish it up, where are we now with these tissue engineering efforts? The best, this is, I'm not showing you the representative work now. This is, but I say it here in big letters, this is the best we've gotten to date in terms of growing human heart muscle back in an injured animal's heart. I can tell, I say with a little bit of pride, I can now show you something with a 2x objective, which means this is really low magnification, right? So and we now have a human myocardium. This is a human cardiac patch sewn to the surface of a rat heart. And the thickness of the human myocardium is equal to the thickness of the rat infarct. It still has this darn band of scar tissue that you see here. And that remains a barrier to electrical integration of this. And it's something that we're working on. But in terms of mass of new myocardium that we've generated, before what we had was little wisps that looked sort of like these little strands of surviving host tissue that you're seeing here. So our best results now are 10 to 50-fold better than what we had using cell therapy 2 years ago. So we're making some progress. This is really low mag. What does this stuff look like? If you take that little red box and blow it up, you get stuff that's looking reasonably mature. This is 3 months after implantation. These cells are starting to slow down in terms of cell division, and they're maturing. So we get nice-looking sarcomeres in some of them, and it looks pretty reasonable, actually.
Dr. Murry, what's the age of the infarct there?
So the way we did this experiment was created the infarct and then came back 4 days later and put the patch on. So it's about a 3-month-old infarct because the patch was left in so it's 3 months and 4 days.
And Chuck, what's that stuff that looks like squamous epithelium there on top?
Oh, stop that, Dan. I think that's actually fibrous tissue, but occasionally we do see little cytokeratin-positive epithelial elements when we haven't cleaned up our preps well enough. And so, purity. I mean, is this ready for prime time? No. Would you want your mother to have this? No. But you might want your nude rat to have this. If your nude rat has an infarct, we can really help it this year. I'm pleased to be able to say that. So this is clearly a garbage in, garbage out kind of phenomenon. When you get really good cell preps, you get great looking results. When you have lousy cell preps, you have problems with heterogeneity and heterologous elements, as we like to call them, because we don't like to use the teratoma word too much because it scares people.
So does the graft contract autonomously?
That's a good question, Andy. I deliberately took that section out because it gets complicated. The question was, does this beat, basically, right? And does it beat autonomously or does it beat in sync with the rest of the heart? And the short answer is we don't know yet. What we know is that in general, at least with the cell therapy approaches, we improve function of the heart, but that could be the host cells that are doing the work. It doesn't prove, we know the graft cells have initiated the repair, but we don't know who's doing the work of systolic contraction. And so the short version is we've built a calcium indicator protein that's a GFP variant that's got a calmodulin domain on it. And so the cells will flash every time they beat. And then we're doing calcium imaging studies, and we can time the beating of the grafts with the beating of the heart, and we'll know whether they're in sync or not. And I think that's about as well as we can do now. And if they're in sync, I think we can conclude that they're probably at least contributing systolic force. But be happy to talk with you more about that. Again, just to finish up, these are, again, best results we've had for vascularization. We're getting pretty good networks of human microvessels in some of these hearts. And you can see these tend to be thin-walled little sinusoidal channels, but lots of red cells in them. So clearly they must have anastomosed to the rat's coronary circulation. So what do we conclude? That if you want to do something like repair a tissue, you've got to take advantage of the fact that tissue have multiple cellular elements that have crosstalk that's necessary for normal homeostasis. A corollary to that really is that if you are focused on just a single cell type, like we were for years just putting myocytes in, you lose a huge amount of synergy that's normally available by crosstalk between different cell types. And Mother Nature is very smart, way smarter than we are. And these cells have this genetically based evolutionary program to self-assemble and undergo this kind of morphogenesis that I think can be very useful in situ if we can just nudge them in the right direction. It's clear that if you add vascular and stromal elements like these mesenchymal cells I was talking about, they have a big impact on human cardiac muscle survival. And I haven't had time to tell you this, but when we get big grafts in the heart, we also affect the coronary collateral circulation. So we actually remodel the arteries that end up feeding into this. And this is something I think is very interesting to follow up on as well. So what's for the future? One person's opinion about this. People often ask, if we're going to repair a heart with stem cells or repair anything with stem cells, what's the best cell? Should we use adult cells? Should we use embryonic cells? Should we use resident cells from the heart? Should we use bone marrow cells, et cetera? I think based on these results that we've had with tissue crosstalk, looking for a single best cell type is probably not a good strategy. I clearly think that it's time for us to shelve this debate of adult cells versus embryonic cells. We know that now that you can take a skin cell and reprogram it back to being the equivalent of an embryonic stem cell, that all of this is going to cease to become meaningful, and I think it should go the way of the Bush administration. I think that we also need, even though it's difficult, we need to embrace complexity. I think these cell-cell interactions are going to be very important. And yes, it's harder to study, but it works. And failure is very depressing. And so I'd rather have success that I didn't understand as well. And the bottom line is that as this gets increasingly complex, it's going to take interdisciplinary scientific teams to start working on some of this. And that's, of course, what we've been trying to do with our efforts down at South Lake Union with the Cardiovascular Program, the Stem Cell Institute, and that kind of thing. And the hope is that, that we'll actually be able to be successful in making some advances that will be clinically relevant with regenerative biology. This is the gang that has done the work that I've been proud to talk about. I tried to mention everybody along the way, so I won't go through things, but this is a really good group of people, and I'm fortunate to have them working with me. And just thanks. The work that we're doing is, we're very fortunate to have a lot of strong collaborators from Rick Young's lab at Harvard, MIT, Mike Rainier's lab, the Vascular Imaging Laboratory, people in bioengineering, and NIH, and a variety of foundations and so forth for the funding. And with that, I'd be happy to take any questions. Thank you for your attention. Yeah.
Question is there any evidence that whatever the poor native regenerative ability of the heart is, that it is related to some sort of stem cell mechanism that's similar to what you're trying to replicate? And what gets me to that is that what's going on with people who are chronic marathon runners and have big troponin and CKMB bumps after these runs, clearly have myocardial injury, Yet they're not all dying of cardiac, of congestive heart failure when they're in their 50s or 40s. But people who have ischemic heart disease for that same period of time do.
Right. So I mean, I'm not going to give you, those are really good questions. I'm not going to give you the answer, as you probably suspected for it. I will tell you there is a great paper coming out in Science today. That's because I reviewed it, and so I know they're just lifting the embargo, where people did radiocarbon dating of cardiomyocyte turnover based on pulse labeling that we all got during the Cold War with atmospheric nuclear testing. And they were able to get birth dates and assess human cardiac muscle turnover. And so check this paper out when it comes out. It's probably available online now if you're that interested. But the bottom line is, in the human heart, there is a little turnover. Not much. The quantitative data suggest about 1% per year at age 20 of your myocytes turnover. And by the time you're 50, it's probably down to about 0.4%. What that means is that if you're a 50-year-old man like I am, or almost, that 55% of my myocytes are the ones I was born with, and 45% were probably born after birth. So what does that mean exactly in terms of is the glass half full, half empty? I mean, what it says is that these cells turn over very slowly. They're tough to replace once they're lost, but there is something going on. If we can learn to harness that, it can probably be used therapeutically. In terms of what's going on with marathon runners and things like that, I mean, I guess when you get a troponin bump, does that mean you've killed your cell, or does that mean you've reversibly injured your cell? I used to think it always meant that the cell was dead. But we're learning more that cells are kind of like gels on the inside, and you can suffer a little hole in your cell membrane, some troponin can leak out, and they may be able to reseal these to some extent too. So maybe this is trauma but not lethal. Do I have data for this? No. Yeah, Ron.
What do you think the mesenchymal cells, the stromal cells, are doing that's enabling blood vessels to grow in your little patches? Do they hang around? Are they still there? Do they disappear?
They hang around. They hang around at least in the patch. We're just testing whether they hang around in a transplanted situation, and I think they principally secrete things that endothelial cells like. The one that works really well makes a ton of VEGF, for example, so that probably makes the endothelium very happy. They also produce matrix molecules that endothelial cells like, and that the one that works well makes a lot of provisional proteoglycan-like molecules that they're like wound healing molecules, and the one that works poorly makes very little of those kinds of provisional molecules. So I think it could be, I expect it will be multifactorial. I don't think we'll be so lucky as to find just a single factor. But we'd like to find what they are, because then maybe, if we can learn how to do some of this and remove the complexity of cells and just do it with proteins or whatever, it may be that much more easy to translate to the clinic. Yeah.
So you showed, to my mind, kind of two studies. One was on using single cells, and the second one was using tissue. Mm-hmm. And after the single cell section, you showed this really cool, the beating heart. So basically, my question is, why pursue the heart and tissue studies if the single cells seem to be working? Why not just redefine your treatment group and then, okay. Yeah.
Yes, I mean, so it's a fair question. Just to repeat it for people who are off site, it's if you can put in a suspension of cells and they work, why to go to all the trouble of an engineered tissue construct that's going to be hard to deliver and things like that? Is that essentially your question? Yeah, no, that's very logical. The answer is in part historical. These have been parallel thrusts in the laboratory for the better part, for over 5 years, and so you never know when something is going to give and when it's not going to give. That's one answer. The second answer is that while cell therapy is working to preserve function, the amount of remuscularization that we get is actually pretty piddly. I didn't give you the quantitative data, but we remuscularize maybe 4% of the infarct on average, and yet we have this big impact on ventricular function. And so my suspicion is maybe it's secreted products that these cells are making that are beneficial to the surrounding tissue in some ways. But my logic tells me if we can actually more fully remuscularize, there should be additional benefit that we can get. And so it's really the desire to do a much better job of replacing the muscle that is leading me to use tissue engineering approaches as opposed to just dispersed cell therapy.
I want to thank Dr. Murry, and if there's additional questions, maybe you can hang around a bit and we can ask him. But thanks for a great talk.
Yeah, thank you.